I'm glad to welcome Gary to talk about issues in computational biology and have fun.
Thank you very much, Ilya, for inviting me out here and actually picking me up and driving me here, which was otherwise would have been hard for me to get out here. But this is a really cool place. It's really fun to visit here and see what you guys are doing. So I'm run a research lab at University of Toronto in the Donnelly Center for Cellular and Biomolecular Research. This is what our building looks like conceptually. So if you're ever in the neighborhood, you can look for this one. Actually, there's other buildings here that the architectural diagram doesn't show. So it's kind of hidden behind other buildings, but it's a college and university. So you guys are welcome to come by and check us out. We have a nice bamboo garden in here that's kind of nice to hang out. So I work in the area of computational biology or bioinformatics. Some of you might have heard or know something about this field. There's a bioinformatics undergraduate program at University of Waterloo that I think has like 80 students a year or something like that. And basically, computational biology seeks to use computers to answer biological questions. And generally, this is becoming more and more interesting in biology and medicine because new high-throughput new technologies are allowing people to generate large amounts of information about biological systems that we really never had before. And this is all stemming from the Human Genome Project that started in the late 80s and 90s. So we have terabytes and petabytes of information, and finally you actually need computers to reason over it. Whereas before it was more like a cottage industry. Everybody in biology, everybody had their favorite gene, and they wanted to know what it did, and they tried to collect as much information about it as they could, and that fit in their head. And now when you have to deal with whole systems and larger amounts of data, you have to use computers. And so the cool thing about it is,
is your laptop?
My laptops should be working. I think it's cycling.
There we go. We're good. Sorry.
Okay, just let me know.
Yeah.
I don't have feedback from if I'm facing this direction. Okay, so computational biology, one of the fun things about it is that you guys, okay, so.
It's the remote site is changing it.
Okay.
I believe.
Okay, well, we'd like to just keep it on the presentation, or whoever's listening. So one of the cool things about computational biology is that it uses information and techniques from all areas of computer science. So there's a lot of work on, and you get to kind of learn and talk to people in all these different areas when you're trying to answer biological questions. So there's a lot of work on algorithms, string processing, for instance, and text mining is string processing when you're looking for patterns in DNA, and aligning strings is very important. Machine learning is extremely important, predicting looking for patterns in large data sets and data mining, statistics, statistical clustering, math is used for simulations, knowledge representation is used. We're trying to construct ontologies that organize all of the knowledge and descriptions of biological systems, databases, and software engineering to create usable applications. Our users are typically biologists, and a lot of biologists actually aren't always computer literate or computer savvy. So there's actually this I think it's changing now, but a lot of people who didn't like math went into biology. And so it was, we have to work towards making usable software that anybody can use. So that's important. And most people in molecular and cellular biology are trying to figure out how this system works, the cell. So we're trying to understand the cell. It's a very complicated system. Many of you probably know about this from high school biology. And we're trying to find out how it works and how it doesn't work in disease. And if we know how it doesn't work, then we could potentially design therapies to fix it. And I'll just say just now that if you guys have any questions about biology and or questions if I'm moving too quickly over certain topics, then just interrupt me and we can have a discussion. I think we have plenty of time should have plenty of time for discussion. So what my lab is interested in is kind of developing a computational map of the cell. It's kind of very similar to Google Earth, but of the cell. And we'd like to develop this, basically take all the information that we know about all the systems in the cell, their geography, their connectivity, how important they are, what their role is in biological processes, and organize it into this computational map. Exactly how we do that, we're not totally sure yet. But once we have this map, we'd like to be able to read it to understand how cellular processes are working, what as I said, the role of specific components in those processes are, what happens when those processes fail, like disease, you have a mutation, and that puts a kink in a specific system, and also study these maps between different organisms. So we take the fish map and the human map and the monkey map and compare them to see what's important. We might be able to see the parts of the map that are important for speech and language and higher reasoning that we exhibit compared to fish or monkeys. So those are the types of things that we're working towards. And we have a few projects. Many other people are working on similar things like this. We work to try and predict this map from the genome. So the genome is all of the DNA that is all of the genes, basically the set of all genes in our body. And some of you have probably heard about the Genome Project that sequenced all of the DNA. It's 3 billion letters of DNA bases, code for about 20,000 genes, and all of those those pieces of DNA are organized into chromosomes, and those are compacted into cells, tiny fraction of a meter in diameter, and it's actually basically 3 gigabytes are present in each cell of information. And the way that biological systems actually encode that information is incredible, because in those 3 gigabytes is enough to specify our bodies. And we have about between 50 and 100 trillion cells in our body. So think of 100 trillion times 3 gigabytes, and you get the amount of information that's sort of just encoded in DNA in our bodies. Actually, it's not exactly 100 trillion. It's not all our cells. A lot of bacteria and other things are in us that also have DNA and contribute a lot to the number of cells that we have in our body. We'd like to be able to read that information and figure out how it encodes the biological processes that make us tick, basically. We'd also like to find out how the map works over time, sort of get a more dynamic view, and I call it the active cell map project. And that is hard to do because you have to look at the system over time and make millions of measurements. That's becoming easier to do as technology allows us to make millions of measurements at the same time on these cells. I won't really go into too much of this technology, but we can talk about it if you guys are interested. And I'll talk during this talk just about 2 projects that we're working on. One is called multiple perturbation mapping of the system. And it's very similar to reverse engineering. I don't use the term reverse engineering because the system wasn't engineered. It's evolved. So instead, we're just perturbing it and seeing what happens. I'll explain how that works. And also talk about some of the software and databases that we're developing to help visualize and analyze this information. So multiple perturbation mapping is basically trying to understand what's happening in the black box. And we have, many of you are probably familiar with this analogy. You have a series of inputs on one side and outputs on the other. In our case, these switches are all on, and this black box is somehow working, and that means this light is on. And so the light's always working, and the system's working, light's on, that's good. But if you turn off one of the switches, the light might turn off, right? And so this tells us that if we turn off switch A, we have some kind of direct effect that somehow A is directly connected to the light. And if we change now, we turn back on A, and the light goes back on, we turn off B, but nothing happens when we turn off B. So is this connected at all to the light? Well, maybe we also see the same thing with C. C, we turn it off and nothing happens to the light. But if we turn off B and C together, then the light turns off, right? So somehow B and C are connected in some parallel path. If you see this type of pattern, you might be able to kind of tell something about what's in the black box. Maybe these, either A or B needs to be functioning for the light to work, and only if they're both off does it fail. So that means you have some kind of parallel pathway that's in that black box, logically at least. And then you try the next switch, D, and also that turns off the light, right? But how does that relate to A turning off the light? So what happens if you switch A? It does, maybe you see that you switch A up and down and nothing happens, right? So that means that D is somehow masking the effect of A. And that probably means that D is kind of upstream of A in some path that allows you to figure out what's going on. So through these switching, all the options, you might be able to actually figure out something about the wiring inside the box, right? Does that make sense to everybody? So our black box is that we're studying is budding yeast. So budding yeast is also known as baker's yeast and brewer's yeast. All of you have eaten this, actually. And this is used, it's also, the Latin name is Saccharomyces cerevisiae. It's a fungus, and doesn't sound too good, but it's actually one of the fungus that's good that we use in industry. It comes from the Greek sugar mold and the Latin of beer. So it's used in the beer industry, and it has 6,000 genes, 6,000 parts. They're organized into 16 chromosomes, and here's a little video that just shows a little bit, yeast under a microscope, and you can just see them kind of growing for a little bit. They're called budding yeast because they grow by budding like a new bud off the side of the yeast, and then once it's fully budded, then it splits off and it becomes its own cell, and yeast budding yeast grow in this manner. There's lots of other types of yeast around that do different things, but these ones work like this. So and if you let them grow, they'll grow into a big colony, and if they have enough food, they'll take over a agar dish or something like that. So they're pretty fun to look at. So why do we study yeast? Well, basically, it's a simple system. We're trying to understand how the human body works, but it's kind of difficult to do experiments on the human body, plus we don't want to. So yeast is a simple version of us. It's just one cell instead of 100 trillion cells or 50 trillion cells. It's easy to work with in the lab. And 20% of the yeast genes are very similar to genes that we have in our own genome. So there's similar processes, there's similar core processes. So yeast have a cell cycle that allows them to grow, and we also have those types of things in our own cells. And a number of famous discoveries have been made in yeast. The Nobel Prize in 2001 went to a number of yeast researchers who used yeast to figure out how cells grow and how the timing is and how that's controlled and how the timing works. And that was very relevant to cancer, it turned out, because cancer is a disease of cell growth. The cells just grow uncontrollably and they take over, right? They're not working in a symbiotic fashion with each other. They're not playing nice with others, basically. They're just growing and taking over all the resources. So understanding more about cell growth tells us a lot about cancer. And if we could understand, for instance, how to stop cell growth, you might have some anti-cancer drugs as well. It's also, other types of fungus are pathogenic. So there's different types of bad yeasts that you don't want to have. And there's important to study yeast to find antifungal drugs. And also, last but not least, the biggest use of yeast is that we actually farm it. We use it to make bread, beer, and wine. And basically, yeast are really good at taking sugar, like glucose, and converting it to ethanol and carbon dioxide through a process of fermentation. And in bread, the alcohol is kind of made, but it's baked away, so you can smell it in the air when you're baking really yeasty bread. But in wine and beer, it's kept, and the carbon dioxide is kept in beer, and it carbonates the beverage, right? It doesn't happen. You can get rid of that if you want. But so where would we be without yeast, right? Italian restaurants and a lot of Irish pubs would not be as exciting. So basically, we'd like to do this black box thing that we were doing before with yeast cells. And instead of having 4 switches in that simple example that I gave you, we have 6,000 genes. So it's a much more complicated system. And we're measuring, instead of whether the light is on or off, whether the cells grow or don't grow. So if the cells grow, the light's on. If the cells don't grow, the light's off. And this is sort of a logo from the yeast genome database, which they have a super yeast, they call it superbud. Anyway, this is their logo. So it turns out that people have done the work to delete each gene individually. So all 6,000 genes have been individually removed, and people from that experiment, which was a pretty big experiment at the time, about 10 or 15 years ago, they figured out that 1,000 of those genes were essential for the yeast to grow, and 5,000 of them, if you took them out, it didn't matter, actually. The yeast would grow. Maybe they wouldn't grow properly in every condition that you threw them at, right? Maybe they wouldn't, certain things would be broken. But just in terms of growth or not growth, it's 1,000 genes are essential for that process. And you can actually download these. All of this information that I'm talking about in biology is pretty much all open access. So anyone can access the information. So the other little difference between that simple black box that I mentioned is that an individual gene is not just a switch that you can switch on and off. I mean, we're removing if we remove the entire gene, it's like we're switching off the gene. But that gene is actually much more complicated. So a gene is encoded in DNA, and it's converted to a protein. And the protein folds up into a little molecular machine. You might have heard of the word enzyme, which is a specific type of protein that does something, breaks things down in our body and builds them back up into muscle, for instance, in metabolic pathways. And proteins connect with a lot of different other things in the cell. They connect with each other. So instead of just having a simple switch, we actually are working with very complicated components and interactions. And there's all sorts of different ways that these can be modified by the cell in fairly complicated processes, which we know something about, but a lot of it we're missing. Like here's an example of two proteins connecting. These are 3D structures of proteins. These are really, really tiny molecules that connect, and they interact in a very specific way. So just to give you an example of the complexity of each gene, what we're doing here is just knocking each gene out. But if we really wanted to understand the system, we'd have to understand a lot more about all of these things. So knocking out individual genes tells us something about how the genes are important in the system, but it doesn't tell us everything about what we want to know. So it's a good first step, and it helps us get some level of mapping of the cell and figuring out what's important. So obviously, you've tried switching off all the genes individually by removing them individually. The next step is to, and we found that there were 5,000 that really didn't have an effect. The next step is to try doubles. So if you take if you remove two genes at the same time, you can see some interesting effects, just like I showed you in the black box example with the upstream effect or the parallel path effect. And we'd like to actually knock out all possible do all possible double gene deletions and see all of these effects. There's 18 million combinations, though, or actually 36 million if you do both directions. And that's a lot of experiments to do. So if you were doing each one of these individually, you'd have to do it 36 million times. That's a lot of effort. So I'll explain how my collaborators in Toronto in our building, our people are working on this problem, how they do it. So, but the basic idea is that you have, similar to the black box example, you knock out, here's an example of one type of interaction. It's the parallel path interaction that I showed you before. You knock out one gene and another gene, and the yeasts are viable, they grow. But if you knock them both out together, then the yeast doesn't grow. And this might indicate, if you do this enough times, it might indicate that if you do this enough time with A and B, you might kind of see pathways or paths through this network of interacting molecules or interacting genes appear. So here, all the yellow lines are this type of parallel path interaction. We call it a synthetic lethal interaction. It's just the jargon of the field, and so it's SL interaction. And these types of white arrows are, connections of in a path, one step, then the next step. And both of these pathways impinge on some kind of essential function, and that then is related to growth. And just by doing all possible combinations of knockouts, you might be able to see that there's actually two pathways and that they're parallel. So that is just an example to tell you how things are working. And it's not just growth or not growth that we're measuring now. So it used to be actually that was the case, but now we're actually have learned how to in the lab measure this quantitatively. So we measure this. So it's not just the light is, on or off. It's it's different types of dimness or brightness. And WT is this sort of the normal yeast. People call it wild type. It's just the type that you see in the wild. And we'd call that 1. That's growing normally. And then you knock out A and you go down to 0.5 level of growth. You knock out B and you get to also 0.5 level of growth. And you knock out A and B together, you expect that there's going to be, because A and B each have an effect, you expect that the A-B combination is going to have a worse effect, right? So we have some expected results. That's a neutral result. That's just the multiplication of A and B, basically. And there's different ways you can figure out what this expected result is, but that's just the expected result. And then you can measure what the actual result is and see if it's less than you expect or more than you expect. So if you get less than you expect, you get one of these synthetic lethal interactions, which gives you an idea of parallel systems, because you can think of it as like the A-B parallel path that I mentioned before. If you have two systems that are parallel, they're backup, they're redundant. If you mess with one of them, the other one takes over, and vice versa. So but you've messed with both of them, then you really get system failure. So it's much more than you would expect, given the changes of the individual perturbations, right? And so that tells you something about the parallel systems. On the other hand, we get alleviating interactions, which are better than you would expect. And these things tell us often about genes who are products in the same type of non-essential system. So if there's some kind of system in the cell that's working, but it's not really essential, then messing with it might affect growth, and messing with it again might affect growth, but not as much as you would think, not as much as you would expect if it wasn't connected. So in those ways, there's three basic types of genetic interactions that we try and map. And we want to do that on a global scale. So as I mentioned, there are 6,000 yeast genes, and 1,000 of them are essential, but that leaves 5,000 non-essential genes. And we'd like to examine all the 36 million combinations, or 18 million if you just do it one way. And Charlie Boone, who is a researcher at University of Toronto, developed over the past decade or so a method to do this experiment in a high-throughput way. So instead of just doing one in one like petri dish or test tube at a time, you can do 1,500 of them on a small plate about this big, and it can be automated as well. So I won't really go through this in too much detail because the biology is really not important. But basically, he's developed a system, and him and his lab has developed a system that allows you to make sure that you combine single deletions to get double deletions. And that's through a series of robotic pinning steps and mating and genetic selection events, which allow only certain type of resistant strains to progress through the process. And if anyone's interested in details, I can get into that. But in the end, you get these double mutants. And this actually led to a network of connections. The first, what you can do with this process is you can take one gene and you can mutate, you can query it against the entire genome, or the rest of the 5,000 genes that are non-essential. So you take one against 5,000, and you see how many of these interactions that you get. And this is plotted here for 8 of these full genome screens that was published in 2001, and a few hundred interactions resulted. And then in 2004, this was expanded to 132 screens, and we got a bigger network. And these networks don't really tell you much. In fact, we usually call these networks hairballs or Death Stars or something, because it's nice to kind of, at this level, to see how things are connected. But when you get too many nodes and edges, you you can't draw it very well. So it's one of the challenges in the field is actually drawing these, building software that can lay these networks out in a reasonable way and interpret them. So I'll talk about that later. So, and this process is being automated by Charlie and a whole team of people. And there's rooms in our building that are just dedicated to robots and arrayers that have plates in big sort of round loaders that you can load in hundreds of plates at a time. And overnight, it just does the experiments. It just does all the pinning. And I have a picture, a movie of one of the older versions of the robots just working here. So there's a plate, it has a cover, it takes the cover off, it takes some pins, and it touches the plate. The plate has, each of those pins is a colony it touches a different colony of yeast growing on the plate. And then it moves the yeast to another plate where it goes through this series of steps that at the end, by the end of a couple of days, you have double mutants growing on the plate, and you can measure their growth rate by looking at how big the colony size is. So if the yeast aren't doing too well, it's a little dot where the pin landed. But if the yeast are growing nicely, then it's a big circle, and the yeast have grown from that pin prick to be a circle. And you can measure that with, you just take a digital image and then process that with image analysis. So this is sort of the output plate that we get. And all the experiments are done in replicate. And there's a number of algorithms that have been developed by the team of people that are working on this project to detect the colonies here with image analysis and count this number of pixels in the colony, and then there's a whole bunch of data correction steps. Actually, I think I should have gotten rid of that slide, but there's a whole bunch of data correction steps that you have to then consider to filter the data. But basically, the thing that we're measuring is the size of these colonies, and that's the and there's a whole bunch of, one of the challenges that we have to face when you have thousands of experiments that you're running is to deal with variation that are due to systematic effects in the some of which are experimental factors, and other that we want to normalize out, and others are biological factors, which is the actual signal that we want to detect when we see an interaction. So all of the, is this clear to everybody so far? Yeah. All of the, this is sort of one genome-wide screen, all the plates that, actually this is I guess probably a couple of genome-wide screens. I think one screen is about 14 plates. So I think that's 14 across. So this is a few screens. This is the images that you get if you look for positive and negative interactions just on the raw data. And all the green guys are positive interactions, and the red guys are negative interactions. But it turns out a lot of these things are there's some artifacts here. So and we've discovered a huge amount just from running so many screens. It's probably very similar to how Google has worked in data centers. When you have huge amounts of hard drives, you start figuring out things about the global properties of hard drives that you didn't know before, right? Because now you've tried it on a million of them. There was I guess Google published a paper on that, right? So similarly, we found that the place that the Boone Lab was getting plates from, one of the tables that they were pouring these gels for the yeast to grow on was slightly lower on one side than the other. So that led to a slight deviation in the amount of food that the yeast were growing on, and there was bigger colonies on one side than the other, right? So, and when we told them, they would have never figured that out because it's, they've calibrated it, but they really didn't know to that level of detail. Other things that, I'll mention some other things in a second, but you get, once you take the raw data, you normalize it, it looks is a lot cleaner. So there's adjustments for the camera that was used. There's plate normalization. So it turns out that the position of the yeast growing on the plates is important. If they're growing at the edge, they get more food because they don't have competition from their neighbors. So the middle ones are growing less on average than the outer ones because they have more competition for more neighbors. And there's the spatial gradient normalization. It's not just that table issue. It's also when they pour this liquid gel into the plate and then it hardens. It's an agar type plate, and the yeast are grown on top of it. The gel is kind of somewhat hard, but depending on where they pour it, there's waves in some way that's somewhat hardened a little bit, and you can detect that and normalize that. There's batches, so there's say 10 different technicians just loading plates into these machines, and each one has their own statistics associated with them, right? They have to normalize different batches, different days. All of these things are things that you would never have to deal with on a small scale typical experiment, but you do when you ramp up to a large scale thing, and Google knows that very well. So in the end, you get an estimate of the mutant, the deletion fitness, and also the double mutant fitness, and you can use that to drive interactions. And you get this score distribution, and you can do more statistics on this. And eventually, with some appropriate cutoffs, you can say there's 45,000 negative interactions and 20,000 positive interactions at 95% confidence. Remember that this is just looking at growth. If we looked at any different type of output, like the size of the cell, or how long it takes to grow, or the maximum density of cells that grows, all of those things are different phenotypes, we call them. They're different outputs, and each one would basically get its own type of network that you'd get. So this is the network of related to growth, and it tells you what's important and what processes are in parallel just for growth. So you can keep on doing this. You could do this experiment with growth on a banana leaf, which is where yeast might have been found from, or in the lab on a plate, or in different conditions, higher temperature, lower temperature, et cetera. So 45,000 interactions doesn't sound a lot out of 3 million gene pairs tested, but it's only one type of context. And here's the whole matrix. You can't even really see it here. We've currently done the Boone Lab, and there's other people who are working on this, like Brenda Andrews, who also works with this team, have done 1,000 of these screens against 5,000. And some people in Princeton, Chad Myers and Olga Troyanskaya's lab at Princeton, have this really nice display wall that we can actually view this thing in. And Anastasia Baryshnikova is a student in my lab, and Charlie Boone's lab that works on this data. And so, when you get to this much data, there's somewhat challenging to display it. And there's certain nice toys that people have that we can use for some of this stuff. But really what you want to do is find out more about how the system is organized. That's what we're interested in. And if you zoom in on this data matrix, you can see regions that are clustered. So this data matrix is clustered. How many people are familiar with clustering? So basically it's just an algorithm. There's lots of different types that put similar things together and separate different things and put them apart. So similar screens are grouped together in this. This is average linkage hierarchical clustering, if you're familiar with it, and also similar screens and similar genes are grouped in 2 dimensions. And so if you zoom in, you see genes that you can find little clusters where all the genes have similar types of activity. And in this case, there's some particular system that's important to the cell for membrane trafficking, which is basically just moving molecules inside and out of the cell. That's one of the systems that's important. There's another little cluster here that tells you something about DNA replication and repair. So one of the cool things about biological systems is that they're self-repairing. We're always repairing ourselves. Cells are falling off and new ones are being generated in our skin. And we can go out in sunlight and not worry about it because UV light that damages DNA is repaired. Any of that DNA damage is generally repaired, usually. And so that's an important system. And you can see also additional patterns that tell us more about how these systems are organized based on whether the interactions are positive or negative. As I mentioned, the positive or green interactions are more within systems, non-essential systems, whereas the negative or strong interactions are between important systems that are buffering each other or redundant. And so you see this, the green guys, all of these, basically each square here represents a measurement for one gene and another gene. And the measurement is the interaction strength of the pair of genes. And so all of these guys, all of these genes here interact with each other positively. So this is a particular system called, biologists have a huge amount of names, by the way, for all sorts of things. So that's one of the things that you have to learn in biology is memorize all this dictionary of things. But there's something called the vacuolar ATPase complex, which has 10 or 15 genes in it, and it's pretty important for maintaining pH and environment within parts of the cell. So that's, all of these things, you can just see those coming out in this clustering. So we know immediately that these guys are part of the same system. So if there was a gene here that we didn't know anything about, and it was part of this complex, this cluster that that we know something about, then we can make a prediction that says that gene is, we think that gene's part of that system. So all of these interactions are telling us something about components of the systems, and also we can see that these two, this other transport complex is, has a lot of negative interactions between these two complexes here, and that tells us that these complexes are probably buffering each other in some way. They're parallel, and you can draw that as a network, and where each node is a gene, and the lines between them are positive or negative interactions with a given strength. And this is already telling us a lot more about yeast than we knew. So we've already seen that this is a system here, and this system this is another system, and these systems are somehow complementary. And you can imagine that if you do this at a higher and higher scale, you can find out quite a lot of information about how the cell is organized. So that's pretty cool for us. We call these monochromatic effects, basically because we see the same color in the same part of the matrix. And you can also look at sort of different levels of a system view. So this is a system level map that's a very high level. It lumps a whole bunch of genes that we know of that are all part of metabolism or transport, or the mitochondria, which is the energy production center of the cell. And we can see how these systems are connected to each other from a very high level. So that's one other thing that we're seeing in these systems, is that there's a lot of hierarchy. So you might have a general system like energy production, but it has a lot of little components within that are each systems themselves, and then those have systems within them. And within each of those levels, you see these types of this type of organization, buffering and parallel components. And our challenge is actually, one of the things that we want to do with this cell map is organize that completely so that we can get an idea about how these systems are organized. And one of the applications that we could have with such a map is to predict what's important. We might be able to predict other doubles or even triples. We might be able to predict that a particular gene, or we might be able to read off the map that a particular gene is important for one system, and another gene is important for another system. And if you remove both of those systems are important for the global process of the cell. So removing important genes in each system, taking down each system, will then take out the whole system, right? And some people have proposed that might be an interesting way of treating cancer. One of the interesting things with cancer is that cancer's 400 different diseases. It's not one disease. There's, breast cancer and lung cancers are very different from each other. And but they obviously have similar outcomes. But one of the other similarities they have at a molecular level is that there's all mutations in cancer. So cancers are somehow abnormal. And if there's some problem in a cancer cell, it's lost a function that allows it to grow. Say it's lost a break in one of these systems, and because it's lost the break, it's going all the time, and it's growing out of control. That differentiates it from normal cells in that it's missing a component. So say we knew something about the way that these systems are working, and we could say, okay, this cancer is missing a component. Why don't I take out this other particular component and this other particular component, and now I will predict that by removing those components, that in addition to the component that it's already missing, takes down this system, right, and kills the cell in a very specific way that doesn't affect normal cells, because normal cells don't have that missing component. They don't have that mutation. So that's very theoretical, but it's one idea that people have in cancer therapy. So just one example of how that might work. Another example that's kind of interesting in the future to talk about what these maps can do for us is designing artificial industrial yeasts that can do new things, right? So we can engineer a yeast cell to do carry out some process that it could not otherwise carry out, and that might be useful for industry, like generating a particular type of chemical. It's already useful for generating alcohol, right? But maybe it could be designed, people do this actually already. They design bacteria and yeast to generate new things, like generate insulin or drugs, other types of drugs. A lot of protein drugs like insulin for diabetes is grown from bacteria in big vats. these days. It's not taken from dog's pancreas, which is where it was originally found. But the problem with building these systems is that these systems that we're engineering are evolving, and they're competing with each other. And if we introduce a system into this, if we introduce a new process into the system, it's not necessarily going to be as fit in terms of that large competing system, right? The large competition of all its neighbors. And maybe it will actually, because it's using up more resources to do something that we want, it won't be able to grow well if another bacteria comes in. It won't be able to grow as well, and the new bacteria will just compete out and eventually take over the whole system. So understanding something about how the system can compensate for how different components of the system can compensate for each other might allow us to build redundancies into these things. So, and also consider what effect our changes would have on growth and other things that maybe you could figure that out through simulations and things. People actually do this that allow you to design a more robust system that would be more robust in terms of the global scheme of things. Here's another view of a systems level map going into a little more detail. So instead of having 10 major categories of gene function, we have a few hundred. And if we look at just sort of very specific ones, you can't really read these, but it just gives you an idea of the hierarchy. We could zoom in on these things and see progressively more resolution on these systems. So that's basically it for the multiple perturbation part of the talk. So these screens are going on 24/7 in the Donnelly Center, and in a couple of years we should actually be able to complete all 18 million experiments. It's probably going to be 36 million because it's easier to just do all of them rather than trying to pick individual ones. It's easier to just do them all. So in a few years, 36 million experiments will be done. Would have taken quite a long time without all the robots. And we should have a much better idea of how yeast work. One of the big challenges to getting towards more applications that I told you about would be to try and replicate this in other organisms and see how well these systems, these maps are conserved. Is this the same kind of map or the same type of properties that we have in our own in human biology? That's an open question that people are starting to work with right now by looking at different organisms. And so there's a lot of work still to be done. But I wanted to switch gears a little bit and tell you something about the software that we're developing to help us develop cell maps, explore them, and analyze them. We have to consider, just like in Google, we have to consider all of the different, we were talking with Ilya earlier and talking about, logs, dealing with logs and analyzing log files, and also, all of the web pages, obviously, that Google is collecting. We have to develop systems to deal with all of this data as well. It's not as much data as Google, but it's still we don't have as many people either working for us. There's information that we'd like to collect from lots of different sources, integrate it together, and then analyze it. And we kind of think about this in terms of I call this the systems biology pyramid, and this is coined by Chris Sander, who is someone I worked with when I was in New York doing research there. And basically, when we're trying to study biological systems computationally, we want to develop computational models that will allow us to predict something about the system, and then basically those predictions can be validated or falsified by experiments that could hopefully allow us to improve the model, refine the model, and eventually the computational model will be telling us something about how the biological system is actually working, right? So that's already what I talked about. But the information system and in building those models, we have to collect all of the data that we can to build them. Otherwise, we're missing information that we've already discovered. And there's lots of different sources for this information. So the information system that we need to build, you guys are very familiar with this type of thing, but you need something to store, organize, search, visualize, and analyze the information. And so we're building this system called Pathway Commons, and this is done with people in New York and Chris Sanders' group and a number of other people worldwide. It turns out that a lot of different people have been developing databases to do this. And so ideally that would actually help us because we could just go to a database and download the data. But because it was such a popular activity, there's actually 250 of them. And that makes it hard. Not mostly because there's each database has been developed independently, and each one uses a different language, different scope, different formats, and so you can't really just collect seamlessly all the information from all the databases. I guess this is somehow similar to this deep web thing, right? So we have that same kind of problem. So one of the things that we've been developing is a standard language that people can use to describe the information, these biological processes, and they represent all the different types of biological processes that we know are important. And it's a community effort, open access community effort that we've been organizing for all these databases to come together and then develop this language, the standard language, that everyone can speak and understand, and distribute. Then they'd be able to distribute their information in a standard format, and then you can start collecting it and using it. The actual format is called Biopax, and it's written in OWL, which is the web ontology language. It's XML. It's a W3C standard. This is an example of what it looks like. I don't know how many people are familiar with semantic web technologies that are being used. We're trying to use that type of technology. The database software that we built some database software as well that allows us to collect, once it's in a standard format, it collects it into a database, typical MySQL database, that makes it available via the web and via a web service so that you can query it, third-party software can query it, and we have a specific type of software that allows us to look at these systems, visualize these systems, and analyze them. And that's called Cytoscape, which I'll mention. And all of these technologies are coming together in this Pathway Commons project that I mentioned. And it's an open access public library of this information. So we're using the analogy of the public library. So these processes, these individual systems that biologists are studying are like books. Each person studies one process in the cell, and they study it for all their lives, and they write lots of papers about it. It's like writing a book. The lingua franca, or the common language, is this Biopax format or other standard formats. We have the database to index it. It's like the index cards, and the translators to translate everything to the common format. And then once it's there, then it's much more accessible, just like public libraries made literature accessible a long time ago. And we also have, once we can collect all this information in a central place, then it doesn't necessarily have to be central, but at least it's conceptually central. It's conceptually use the same standard language. We can visualize it and analyze it. And I work on a open source software project called Cytoscape, which is originally developed in the Institute of Systems Biology in Seattle and now involves 9 academic and industry groups. This summer we actually had funding from Google Summer of Code, so we had 9 Google Summer of Code students working on this software and related software, and that was pretty fun. And basically it allows you to load up these networks, load up a lot of different types of information on the networks, lay them out automatically, visualize them, and do a lot of types of analysis that people are interested in. Automatic literature mining. So there's tons of publications for this is just one example. There's lots of publications out there that biologists over the past 100 years have published, and a number of those are made available electronically, at least in the abstract form, on the PubMed library from the National Library of Medicine in the US. And those abstracts we can extract information about gene interactions and view them in this software. So that's one of the things that it can do. So that's a way for people to kind of extract information from literature more easily than just reading it. Obviously, it has some false positive rate, for instance, but it's a useful starting point for many people. So, and the challenges for just maybe some of you guys are interested in this, the challenges that we face in dealing with this large amounts of information that are generated by scientists all over the world in all sorts of different non-standard formats is basically how to collect all of that information. So the web is kind of nice because at least there's HTTP. You can download content, but then there's no real structure within those web pages. We don't even have HTTP. Our HTTP is the English language. So maybe it's available electronically. Maybe it's available. It's not even available electronically. The electronic stuff is not standardized in any way. Often, a lot of computational biology and bioinformatics people work in this area just to standardize and develop languages, common languages for biologists to use so that it would be easier to work with the information and to combine the information. And so one of the challenges is to develop systems that integrate with how biologists work so that they can type in their data into the global knowledge base. And that's very difficult because biologists are very busy people, like all scientists and many other people, and they're not interested in doing work for other people necessarily. But if you make it really easy and you give them some advantage, then that might work. So building such a system like that would be very, very interesting and challenging. We have to do integration from different types of data. So if someone says this is gene A in one database and someone says this is gene ABC in another database, maybe those two genes are actually the same gene, but they have different names because people just name them differently. And there's no standard naming system even for some organisms. So we have to recognize those things and pull them together and look for structural similarity in data structures. We have a lot of problems with visualization. Google Earth is really nice because you have we already know how the Earth is a sphere pretty much, and you can map everything onto that sphere. And we don't really know yet we don't have that kind of thing to guide us in terms of laying out these networks. There's a huge amount of information, and we have to somehow make it very aesthetically pleasing. This is already done in textbooks, so biologists are very used to this. If you ever took biology, you'll know, you looking through a biology textbook, there's lots of nice pictures of biological systems, but those were done by artists, and it's hard for a computer to do the same thing. So some automated way of doing that would allow, really engage scientists and do wonders for communication. There's a lot of other things that are important in terms of algorithms and other representations in this way. So those are the kinds of things that we work on the infrastructure side when we're developing software. And we really want to be with visualization with this cell map is, we want the hitchhiker's guide to the cell, right? So I teach some graduate and undergraduate courses at the University of Toronto, and I recently used this analogy of the Hitchhiker's Guide, and none of the undergraduates knew what I was talking about. So hopefully you guys remember the Hitchhiker's Guide. I think for the next course I'm going to have to say the iPod or Guide to the Cell or the iPhone Guide to the Cell or something. But maybe they know Google Earth. Maybe that would be a good analogy. So anyway, it shows my age. But where we really want to be with visualization is this movie, The Inner Life of the Cell, which was let me just stop this here and give it an introduction. There was a movie produced by professional artists who were hired by some undergraduate biology instructors at Harvard, and they kind of made this view of how the cell works internally. And I know some of you might have seen this. How many people have seen this video, The Inner Life of the Cell? Okay, so it's being distributed widely, but it was even on Slashdot, I think. But I'll just show this to you and kind of give you an idea of the complexity of the system that we're working with. It's a very simplified view of what's going on in biology, but they've tried to be as realistic as possible. And so each little molecule was actually taken from real molecular structures, and the shapes and sizes and relative proportions of everything was taken from real factual things that people have found out by looking at cells for many years. And this basically goes through the little story of a white blood cell in your body, which is involved in healing and protecting you from bacteria and viruses, moving into a site of infection and doing something, becoming activated and then moving out. So I'll just show you this video here. So you have the white blood cell moving through the artery, and then it kind of knows where to go by connecting with little molecules onto the blood vessel wall, and recruiting on its surface a lot of different types of molecules that work with cell-cell interaction. So cells communicate with each other through these molecules, and then this movie kind of zooms into inside the cell. So now we're inside the cell. The cell has a membrane, like a bag around it that pretty much is a lipid membrane, and you kind of see this view of the cytoskeleton, which is the thing that gives the cell its structure. And there's lots of different molecules that form like this to give the cell structure, and this is how the cell moves. It creates structure on one side and destroys structure on another, so it's like kind of like a tank, moving things from one side to the other. These are actual proteins that microtubules and actin filaments and things like this and this is how people think they actually form and work in the cell. So this is a motor protein that actually people think really walks like this along these microtubules carrying cargo. These are other systems in the cell that are involved in growth or energy production. This is the process of protein synthesis. So the DNA is translated to RNA, which is then read by this ribosome. It's kind of like the compiler to generate the protein, which is the active running program. And then these proteins interact with each other and go and do something. And this video shows a lot of space in the cell. Really, the space is really filled with these things. There's no free-floating space in the cell. This is sort of how people think these lipid membranes or these fatty membranes work. If people are familiar with biology, this is the Golgi apparatus. So it's very fluid. And then this cargo is released. And these might be molecules that are important for activity at the site of infection, for instance, in this particular case. Once they're released, some of them go into the bloodstream and others are kept on the surface of the cell and then activated at a specific time in order to, say, stop. And the cell rolls along and then eventually stops, and then it knows it's kind of being targeted towards a site of infection. So this is sort of the end of the video. Where the cell actually moves through the blood vessel to its site of infection. You can see the other blood cells there. So this was a pretty cool video that I like. So with that, that's where we'd love to be in the future. I don't know how long it's going to take us to. That was done by artists, but it would be great if the computer could really work like that, and we could have some kind of video game for cells that allowed us to organize all the information and be able to simulate what happens when we do things to it. So the genetic interactions were all done in the lab of Charlie Boone and Brenda Andrews at University of Toronto, and there's a lot of other collaborators that are part of this project in multiple labs. My lab works on a number of other things, and there's lots of people around that do software development in different places around mostly North America and Europe, and some in Japan. And we get funding from federal funding agencies like the Canadian Institutes of Health Research and Genome Canada and those types of things. There's government money. Your tax dollars actually go towards this. My tax dollars too. And we have to, that's how we fund our research, basically applying for grants. So with that, thanks again for listening, and hopefully that was interesting.